welcome back to Fossil Friday Chats. I'm Brittany Sternberg, the Outreach and Communications Coordinator from the Western Science Center. With me, as always, is Gabriel Santos from the Alf.
Hey everybody, good to be back.
And joining us today is Dr. Aly Baumgartner. Hey, Aly, how you doing?
Hello, I'm fantastic. I'm so glad to be here.
We are glad to have you. I remember last season in Fossil Friday Chats, we got a lot of people who had requests for paleobotany. So just wanted to let everybody know we heard your call.
I was really wanting it. I
Yeah.
love paleobotany. I don't really get a chance to talk about it or do it that often. Even when I was doing my master's thesis, I was like, look, there were like some plant stuff that were popping up, and I was like, that's cool, but I have no idea what to do with that. So I'm sorry, plants.
And I appreciate that. I would much rather you be like, I respect you, plants. I will save you for someone else who can actually take care of you. Because when animal people just assume they can do things with plants, that's when I get angry.
Oh, Allie, I'm gonna let you know. We just got a request at the Alf Museum to accession plant material from LA, and other
Uh-huh.
museums were like, I don't know if we can hold it, and we were like, we'll take it, and I was like, yes, more plants.
More
Yes!
plants.
I love it. That makes me so happy.
Yeah, so if anybody's not familiar with Aly and her work, she is the Paleontology Collections Manager at the Sternberg Museum of Natural History. She's a paleobotanist, but took the long way to get there. She earned her BS in environmental science from the University of Michigan and thought she would be a vertebrate paleontologist. However, she fell in love with plants during her MS in biology at East Tennessee State University and her PhD in geology at Baylor University, and now she speaks the fossil trees. I love that.
Yeah, I'm so
Yeah,
excited.
yeah. I guess there's a reason that your Twitter and social media handles are Paleo Lorax, huh?
Oh yes. I don't know. It came to me one day and I was like, this is perfect. This is perfect. This needs to be all of them because I used to have a split brand and that's no good. So now I have a better brand, I guess.
Consistency is key.
Yes,
I felt
exactly.
so stupid when we met like online for the first time when we did like the tour, and
Mhm.
then like when you said it, it clicked in my head. I was like, oh my god, of course it is.
That's why you really need to capitalize it because otherwise there's just a lot of like L's and vowels in the middle and it's overwhelming. So like capitalization is very important.
Well,
As
like
a
a
as
lot
an
of
English
paleo
major, I endorse that.
Well, like a lot of paleo folks like use their name as like some
Yeah.
genus or something. So stupidly, I was just thinking, oh, it's some like plant genus, and I was like,
Yes.
okay, whatever.
That would be
Like,
such a good genus, though. Like, is it?
Allie, remember that for a while. Remember that for the future.
I wonder. Like, has someone must have done that already? Like,
Oh,
someone must have done that
you're
already.
gonna
I would
you're
hope
gonna
so.
have to find out. All right, everybody. If you have any questions for Allie over the course of her talk, go ahead and put those in the chat. We're going to have a Q&A portion, as always, after her talk. So, Allie, whenever you are ready, go ahead and take it away and tell us about plants.
All right. Yeah, so excited. All right, can you hear me? Are there words? Words and sounds? Okay, we good? All right. The beginning? I can do that. All right. Oh, no, it's okay. All right. We good? Let's go. All right. Cool, cool, cool. That was exciting. I'm still a time traveler. See, we're going back in time as we speak. So I'm going to be talking to you about my favorite time machines, plants. So like I said, I am a time traveler, and all of the work that I do for my time travel involves plants. So when I time travel, there are three things that I need to know about any given fossil locality. First of all, what was the climate? Second of all, what was the environment? And third of all, who lived here? All right, so plants are my time machine of choice, and I want to make sure before we go any further that we're all on the same page with what is a plant. So broadly speaking, a plant is a multicellular organism, so like us. They are photoautotrophic, meaning that they use light from the sun to make their own energy, unlike us who have to eat other things in order to make energy. They're also non-modal. So that means that plants aren't just walking around. So with the exception of growth or dispersal, so dispersing their fruits and seeds and pollen, plants are pretty much stuck where they are, and that's very useful. So broadly speaking, plants fall into two different categories: vascular and non-vascular, and seed and non-seed. So vascular plants are plants with tubes in them. So that means that they have roots to suck up water and move nutrients through the plant. So they have xylem and phloem to move everything around. And so that means they can be a little bit further from the water. They can go down deeper to get hard to reach nutrients. Seed and non-seed plants are pretty much what it sounds like. A seed plant has seeds, and a non-seed plant reproduces with spores. So, broadly speaking, non-vascular, non-seed plants include mosses, liverworts, hornworts, things that are very low to the ground. They tend to live pretty close to water. Vascular non-seed plants means that they have roots, they can grow a little bit taller, they can go a little bit further from the water, but they reproduce with spores. So that includes things like ferns and club mosses. And then finally, you have the vascular seed plants. So these are things that have tubes inside, they have roots, they can grow pretty big, but they also have seeds, meaning that they can get a little bit further from the water in drier environments and still be able to reproduce. So that includes gymnosperms, so those are conifers, pine trees, things like that, and angiosperms, which are your flowering plants. And before I move on, I just want to draw attention to the best picture of a plant I have ever taken. This is a baby fern that is unfurling at the Isabella plantation just in London. So most beautiful picture I've ever taken. So I explained what a plant was, but now I want to explain to you why plants are the best time machines. Basically, everything that makes a plant makes it a good time machine. So first of all, plants are non-modal. They're not going to move anywhere. So that means that if the climate or the environment changes, they have two options. They can adapt or they can die. And you might say, Aly, that's true for animals too. And I would disagree with you, because in the lifetime of a single animal, they can move. They can walk away. Plants do not have that option. They cannot try to disperse until the next generation. And that means that plants are fantastic local indicators. If a plant is living in an environment, you can use it to understand what the environment was like at that time. Secondly, plants have fantastic preservation potential. They can lose bits of them without dying. I mean, think about it. They can drop branches and fruits and leaves without dying. Think about deciduous trees every year. They drop all of these leaves and then they come back. That means that plants have many more opportunities for parts of them to be preserved. Wood, everyone has heard of petrified wood. There are national parks dedicated to the fact that petrified wood is very common. Pollen is famously one of the strongest biological substances on the planet. And people tend to think of leaves as being flimsy and maybe bad fossils, but they're much more common in the fossil record than you would think. Honestly, the main reason I think that people think that leaves are not common fossils is because they're not preserved with vertebrates, and that's what people are most focused on. And finally, plants are photoautotrophs. That means that they make their own food. They're using the light from the sun. They're using water brought in from their roots as well as nutrients. That means that they are very closely tied to the climate and the environment that they live in. Therefore, they make fantastic time machines. So I'm going to combine the first two criteria, what was the climate and what was the environment, together because they are very closely related to each other. Because when we think about environment, we're talking about plants. One of the things I like to tell people is that animals live in the environment. Plants are the environment. So if you look at a picture of the world, you can see plants in a way that isn't possible with animals. You can see these different biomes, these different types of environments by these dark green areas on the planet. And so the way that paleobotanists like myself will try to get a sense of what the climate and the environment was like in these fossil localities is by looking at the relationship between plants and temperature and precipitation. So again, I really want to emphasize when you look at this diagram, so this is called a Whittaker climate diagram, you're looking at the relationship between temperature and precipitation. But if you look at the names of these biomes, they are named for the plants. I mean, grassland. Grass is right in the name. Forest, savanna. These are all biomes that are really described by the plants that you find there. So as you have increasing mean annual temperature, you're going to find more and more larger plants, and increasing annual precipitation. Again, you're going to be able to withstand these larger plants. You can have a bigger plant community. You can have higher diversity. So the way that I understand this using plants, I will say up front, I know that there's more to plants than leaves, but that's the part that I focus on. So I live in this nice little Venn diagram right in the center in plant morphology. I dabble a little bit in plant physiology, so how do the plants actually work, and a little bit in plant ecology, who's living there, what types of things are living there, but mostly I'm just a nerd about leaf shape. So let's talk a little bit more about leaf shape. So this is something that you can notice yourself. This is something that anybody can go out and see right now. If you look at these two leaves, immediately we can tell that they're different. So these are cleared and stained modern leaves. And you can see, just glancing at them, that they look very different. So if you start on the leaf on the right, you can see that it is this light pink color, and it has these kind of crinkly edges. So I would call that a toothed margin. The edges of this leaf are toothed. So you tend to have a higher proportion of plant species with toothed margins in colder places. So think of Canada. Canada is a cold place, and they put a maple leaf on their flag. Maple leaves are toothed leaves. So in general, the more taxa that you have, the more species that you have with toothed leaves, the colder the environment likely is. And in addition, I mentioned that this is a light pink leaf. What that tells us from clearing and staining is that this is a relatively thin leaf. So if you think about deciduous trees, they only have their leaves for a growing season, and then they drop them again at the end of the growing season. So a tree isn't going to invest a lot of energy and resources in a leaf that it's just going to drop in a few months. So they tend to be relatively thin. So by looking at this leaf, we can tell that it's thin, so it's probably deciduous, and it has these crinkly edges, so it could live someplace that's relatively cool. Then, if you look at the leaf on the left, you can see, all right, everything is different. So it has these smooth margins. Again, you tend to see a higher proportion of plant species with smooth margin leaves in warmer environments. If you've ever been down to Texas or Louisiana and seen a live oak, live oaks tend to have these very smooth leaves. They also tend to have very thick leaves, and that's what we're seeing here. Because it's a darker color, that means it's a thicker leaf. These leaves tend to stick around for a little bit longer. They're not being dropped every year. So if you've ever seen like a hefty magnolia leaf, those things are kind of leathery. They're pretty intense because they stick around for longer. The plant can invest a little bit more in them. It's important to understand, and I will bring this up again later, that the whole point of leaves is for plants to photosynthesize, and everything they're doing is trying to maximize their ability to photosynthesize. So one of the things that I can do, and I have done at outreach events, and you could probably do it too, is if you can determine the proportion of species in a forest, in a flora, without teeth, and you plug it into this equation, it will give you an estimate for the mean annual temperature. So in general, the higher the proportion of untoothed species in a flora, the higher the temperature. And so this is really cool. This means that all I have to do, I can do this with modern leaves, I can do this with fossil leaves. As long as I can see if they have teeth or not, I can get a sense of the temperature. You could do the same thing with leaf size and precipitation. So, like I said, the whole point of a leaf is to photosynthesize. That's what they're there for. And so plants need to maximize their ability to photosynthesize. They want to have the biggest leaf they possibly can. However, in order to photosynthesize, the leaves will take in CO2. But in the process of taking in CO2, water can escape. This is called evapotranspiration. And if you're in a dry environment, the plants will lose water faster than they can bring in CO2, and that's bad. That's a good way to get dead. So that means that plants will have the biggest leaf they can in order to photosynthesize without being too big that they dry out. So if you look at the leaf on the left that David Attenborough is hugging there, that's a massive leaf, which automatically should have little bells going off in your brain. If you can have a leaf that big, this is an incredibly wet environment because the plant clearly has no concern over losing its water to the atmosphere. On the opposite end of the spectrum, the picture on the right, that is a euphorbia that I photographed in Nairobi National Park in central Kenya. So euphorbia are basically the African equivalent of cacti. So just like cacti, many of them have reduced their leaves into needles so that they can't lose water that way. And instead, the branches in the body of this plant are green in order to photosynthesize. They've basically completely minimized their leaves entirely in order to not lose water. So we're looking at the two extremes here. And just like we could plug in the proportion of untoothed leaves to get the temperature, we can look at the average leaf size in a flora and get a sense of the precipitation. So this is a little bit more complicated. It's not quite as direct, but by and large, if you can figure out the average leaf size, you can get a good sense of the average precipitation. And then finally, my third criteria, who lived here? So in order to explain this, I'm going to tell you a story about how paleobotany fieldwork is very different from vertebrate paleontology fieldwork. So I've done both. And in vertebrate paleontology, there's a lot of just staring at your feet and looking down on the ground for cool looking bones. And if you find a cool looking bone, maybe you can find more cool bones around it, or maybe you'll look and try to figure out where in the wall that cool bone fell from. That is very much not the case with paleobotany. So in paleobotany, most leaf impressions, and a lot of times we're looking for leaves, most leaf impressions are just impressions or carbonization. And so if they weather out of the rock wall, they're just going to be destroyed, and you're probably not even going to notice them. So instead, what we have to do is get very good at recognizing the patterns in the rock layers that normally indicate that here there be leaves. And then we do manual labor. So most of what we do in the field is just go to town on rocks with pickaxes. And as my colleague, Dr. Peter Wilf, once said, you can't get big leaves out of small rocks. So if you are working in an environment that has very large leaves, you're going to need to get really large blocks of rock out in order to get those leaves fully preserved. So yeah, that's exactly what we do. We find the correct layer, go to town with a pickaxe to bring out these big blocks, and then we'll turn them on their side and tap it with a hammer, and it will split along the point of weakness, which is normally where the leaves are. So it's a lot of work. It's a lot of manual labor. And our sample sizes are pretty big. So once we've collected the leaves, then we will take them back to camp. So the way that we do this is we will take the fossil leaves and we will wrap them in toilet paper, and then we will masking tape them shut. When I was doing my fieldwork in Kenya, we would take them away from our field site in these plastic basins, and then we would load them up in the Land Rover. So we were also working with vertebrate paleontologists, but by doing this sort of fieldwork and excavation, we can get a sense of what plants were living here, what animals were living here, and how did they all relate to each other. So I'm going to give you a concrete example of a time that we did that. And I think that this is a really good way of interdisciplinary and multi-proxy approaches to really be a good time traveler and to get a pretty complete sense of what was going on. So this is a paper that actually came out, I think, 2 weeks ago from my dissertation. So I was looking at this fossil site in Western Kenya, so Rusinga Island, which is a relatively small island in Lake Victoria. So I was interested in the early Miocene sediment, so roughly 18-ish million years ago. So Rusinga Island is highlighted in yellow. It's about 10 miles long and about 3 miles wide, and is now in Lake Victoria, near the flanks of the extinct Kisangiri volcano. However, there are two things you need to consider. First of all, 20 million years ago, there was no Lake Victoria, so this was just part of the greater region. And second of all, that volcano was not extinct. That was an active volcano, which is going to be an influence on the local flora and fauna, and also makes it much easier to date things because of the ash. Thank you, Kisangiri volcano. So this locality is absolutely fantastic. So there have been fossil collection going on since the 1930s. Most of it has been focused on the vertebrates. So more than 75,000 fossils have been collected since the 1930s, and more than 100 species of mammal have been described. And they range from huge anthracotheres to terrifying creodonts, early hippo relatives, but most importantly, there are early hominids. So akembo used to be part of proconsul, but now slightly different. And so what I was trying to get at, and the whole reason we did this study, was trying to understand the context for the evolution of our early human ancestors, because if we don't understand where they were living and why and how they were living, we can't really understand why they changed in the ways they did. And so in order to do that, previously people had reconstructed the paleo environment. And unfortunately, it kind of ran the gamut. So it had been reconstructed from everything from a closed canopy tropical rainforest to a mixture of woodland and forest habitats to open and semi-arid environments to a mixture of open and closed habitats. So basically, all of the options for a tropical environment. And so what we did was we looked at 3 floral localities. We were just going to look at the plants in order to get a sense of what the plants were telling us about the environment and how it may have changed through time. So these 3 sites, the oldest was about 18.3 million years old. It hadn't been worked on previously, mostly because there were no vertebrates known from the site. The other sites I looked at, there was R5 that was about 18.1 million years old. I didn't do any additional collections here. This was just comparing to previously published work, but there are definitely vertebrates from that site. And then finally, the youngest site that we looked at is R3. It's about 17.8 million years old. R3 is, I think, the gold standard in like time traveler, being a good time traveler, but we'll get to that in a second. So I collected hundreds of leaves from those localities. I looked at the size and the shape, and I was able to get a sense of the climate there. So as you can see, we've got not much variability in temperature, but a lot of variability in precipitation. And this is pretty standard in the tropics, that it's pretty universally warm there because it was on the equator even 18 million years ago. But through time, we had changes in precipitation going from somewhere between a tropical rainforest to tropical seasonal forest with the oldest site, then getting much drier, being a squarely woodland site at R5, and then R3, the youngest site, was somewhere in between, probably pretty solidly a tropical seasonal forest. But there were other things that we could look at too, not just leaf size and leaf shape. So as I mentioned before, you can look at the thickness of leaves in order to figure out, is this a deciduous or an evergreen locality? Are the leaves, the trees going to be dropping their leaves? And so by looking at and measuring the thickness of my fossil leaves and comparing them to modern forests across tropical Africa, we were able to, again, learn a little bit more about these environments. Kiahara Hill, the tropical rainforest, tropical seasonal forest, if you look at the thickness of the leaves, it's actually, patterns are fairly similar to what we see in other tropical rainforests and tropical seasonal forests in Africa today. If we look at R3, which I reconstructed as pretty solidly a tropical seasonal forest, again, it is very similar to other tropical seasonal forests that we see today, as well as some woodlands. And so if we take all of that together, we can actually get a really good idea of what this site was probably like. So Kiahara Hill had these very large leaves without any teeth. We had evidence of branch casts, meaning that this was probably not just a forest, but a multi-storied forest that was warm and wet, and probably looked very similar to a lot of tropical rainforests that we see across tropical Africa today. As we continue forward in time, we go to R5 and we see something completely different. If you look at the fossil plants and the geology, the sedimentology, as well as the vertebrate paleontology, we get a pretty good sense of what was going on here. If you look at the leaves, we have evidence of cattails and sedges, things that would have been maybe next to water, as well as other relatively small leaves without any teeth, meaning that it was probably warm and not that wet. The geology has evidence of fluvial indicators, meaning that there was probably flowing water, at least periodically, but there was also evaporitics, showing that that water dried up. There were multiple primates found from this site, including a bone bed of crocodilians. So we know there was enough water that the crocodiles were happy to live here, as well as multiple primates that were found. So we know that there was probably a pretty warm, relatively dry, and seasonal environment here that probably looked maybe like this. I absolutely love this reconstruction. It looks so miserable. This looks very uncomfortable. The skeleton there, I think, is a very good touch. And then finally, you have R3, which I think is the best example of combining all of these things to be a good time traveler. Because if you look at the fossil animals You have evidence of things that lived in a forest. You have primates, you have anthracotheres, you got a bunch of cool stuff. If you look at the paleobotany, there were these huge stumps that were preserved in situ, meaning in live position, with extensive root systems. And so they were able to use forestry techniques in order to reconstruct the size and whether or not this was a single canopy or a multi-story canopy. And they showed that this was probably a multi-story canopy. The leaves that I found here were a little bit smaller and they tended to have more teeth. So it was a little bit drier and a little bit cooler than some of the other places, say Kihara Hill, our tropical rainforest. And then one of the coolest things was just looking at the landscape. So where we found these different fossil leaves varied. There were parts of the landscape where all we found were the leaves of these trees. These tended to be closer to the stumps. But if you went a little bit further away, you would find evidence of sedges, which are like grasses, or emergent aquatics. So these are plants that are rooted beneath the water and the leaves are found above. So there was at least periodic ponding on the landscape. So we know that this was a heterogeneous landscape with water and trees, and all of this is supported by what we see with the paleosols, with the fossil soils. And so this is an artist's reconstruction of what the site might have looked like. And I think this is amazing because we're able to take all of these different fields and get a good idea of what was going on and get an actually very beautiful and very scientifically useful picture. So to kind of summarize that, as we move through time, we go from a tropical rainforest drying into a woodland, and then getting wetter again in a tropical seasonal forest. So I think that's a good example of what I do as a time traveler. But the story that I told you was too nice. It was too easy, too clean, and I don't want to lie to you. It can be much harder than that. So I really like this Venn diagram. I think it really illustrates how complicated being a time traveler can really be. So there are so many things that we really should try to consider. Phylogenetic data, meaning the relationships of these organisms to each other. The spatial data, where are these found? The ecological data, how do these things relate to each other? And also the developmental data, how do these things change as they grow? So I like to live right here in what I am calling historical community analysis. I might change the name, but I think it's a good one. Because I told you all of these nice stories about being able to look at the leaves and plug them into an equation and then figure out what the temperature and precipitation was. And it's slightly more complicated than that, which is a good thing. It's always exciting when we find out that stories are more complicated than we thought. Because the equation that I use is based on this global data set. But if you break it up a little bit more, we see these really cool patterns. So the black line is the global number. But if you look at the pink circles in the red line, those are if you just include the data from the northern hemisphere, from Laurasia, so North America and Eurasia minus India. And as you can see, that they're pretty similar. They're not really that different. But if you look at the blue triangles and that light blue line, that's very different. And that's what happens if you look at the plants that are just from the southern hemisphere, from Gondwana. So South America, Africa, India, Antarctica, Australia. These different locations have very different relationships between the leaves and their climate. And that's something that more people should be looking into. And that's where I want to go next. So I think that's the next step. There are so many things that we really need to look into. And there's more to it. What role does soil play? Plants are living in the soil, so we can't ignore it. What about seasonality? An average number doesn't tell you how things are going to be changing. What role does phylogeny play? Does it matter what these things are related to? There are some groups of plants that will always have teeth, no matter where they live. And there are some groups of plants that will never have teeth, no matter where they live. Why might that be? And what about different regions? Does do African floras behave differently than South American floras to European floras? Who knows? These are things that people should really be looking into. And don't worry, I'm trying. And so finally, I have so many people that I should thank. My fantastic field assistants, Suleiman and Mika, they moved mountains for me, literally and figuratively, and I could not have done this without them. And then my many collaborators, my army of undergraduates who helped me process all of my leaves, and all of the people who let me go wandering around in their collections. So thank you very much.
So cool.
Oh my gosh. Thank you, Aly. That was awesome.
I'm glad you liked it. I got really excited. I forgot I was talking to people.
That was so cool. There's like in the over quarantine, as a lot of people our age have gotten into plants, I've been very interested in asking a lot of those questions myself that you just answered for me. So that was amazing. And now my head is like going crazy with questions. So I'm just going to make the announcement now that I will be starting a PhD in paleobotany one day soon.
Yes!
This is Gabe's paleobotany origin story.
I am excited to be a part of it. Like, I want that to happen.
I actually think I will start looking more into it because, like, you know, when I look at all the random plants I have in my room right now, I just like wonder, like, why, like, a lot of questions. Why, like, one question is why doesn't my monstera leaves get bigger? But seriously, like, there's so many things. Like, why does this have holes in it? Like, why are these orchids epiphytic? And all these kinds of questions, and how they survive and interplay with their environment are just so cool. And the way that you explain it from a geological perspective is combines everything I love into one.
Yeah, I was explaining this to someone that I came from a biology background. Like, I did ecology and that sort of stuff. And ecology was one of my favorite classes I've ever taken until I started my PhD and took stratigraphy. And all of a sudden, it was like, this makes so much more sense. Because the things that I was kind of lacking in ecology at the time, that was taken care of with stratigraphy. And I was like, we need both of these things. Like, by combining this, we can get a much better sense of, like, what is actually going on. And I think it's amazing.
It's definitely the same for me. My undergraduate degree is in biological sciences. Then I got a geology degree. And once the two, like, connected with each other, it was just like, oh, wow.
It all makes sense now. Yeah, it's great.
Yeah, I was telling Aly beforehand that I was like, well, I'm getting much more interested in paleobotany, so I might have to ask you some questions because my degree is in environmental studies, and so I'm just very interested in looking at paleo environments. But that's really hard to do when you're only, like, you know, I'm a vertebrate paleontologist right now, so my only reference point has been vertebrates, and they can only tell you so much.
Exactly. So hold on, I want to tell you a story related to that real quick. I applied for a postdoc. I didn't get it, but I applied for a postdoc, and my cover letter was so sassy because I started off with, like, plants are not just the green background, and, like, going off on how you just can't use vertebrates to reconstruct environment because they live in the environment, but they can leave. And, like, goats live in trees, y'all. Like, you can't just use animals to figure out the environment. Like, we should work together, yes, but, like, you're missing something, and don't forget about the plants.
Well, now I'm rethinking my master's thesis because I was trying to do paleoecological reconstructions using vertebrate mammals from a site. So now I'm like,
you can do
brought
that
to the light,
too.
Aly.
I think there were plants for my site, though.
Oh, okay, hold up. If you have plants, there's no excuse not to use them. Because one of the things that needs to be done more, and kind of the project that I was pitching, is basically checking to see if you get the same signal from the vertebrates and from the plants, because that would make it a little bit easier to get away with using animals if you can drown truth it in a place of both.
That makes sense. And all it does is just, like, really emphasize, or what's the word I'm looking for? Like, just back up your hypothesis when you get it from two separate
Exactly.
data sets like that. So I think that's fantastic. I'm definitely, you know, that's a whole other story. I'm not going to get back on my thesis for a while, so let's talk about something else.
That's fine. Hey, if you ever have questions, like, more than happy to talk about it.
Let's see. Yeah, I have a question. So I'm just curious, like, and you kind of already answered it, but like, how would you say that vertebrate paleontologists and paleobotanists should be working together in the future? Like, how would you, like, in what ways would you say that we need to emphasize that? Because I think you're right in that never a lot of times we just kind of focus on our own like study subject and don't go very interdisciplinary. So I would love to hear you talk more about how we can encourage interdisciplinary thought in our field.
Yeah, y'all need to make friends with paleobotanists. Like, that's really where it starts. But no, really, so I was very fortunate. The project that I was a part of in Kenya was part of a larger project in Western Kenya and Eastern Uganda, looking at these early Miocene sites with these early hominids. And it was absolutely fascinating to me that, like, when I think of these localities, I think very like plant focused. Lies, plant focused. But everyone, like you said, comes at it from the direction that they know, and it's fascinating because for that reason, there's just this assumption that like, oh well, there are no plants. I didn't find them. There are no plants. We don't need to talk to the paleobotanists because like, what are they gonna do? And that's so not true. I can name multiple projects, like right off the top of my head, where vertebrate paleontologists tried to bring in paleobotanists, submitted these big grants, and the funding agencies, when they would read them over, was like, there are no plants there. Like, well, no, there are, I swear. But yeah, like just start talking to paleobotanists, start talking, and not just paleobotany. I'm also going to bring in paleopodology. You need people who work on soil because soil and plants are really the way that you get at paleo environment. And people know that soils are preserved. Like paleontologists believe that paleosols are a thing. But leaves are actually typically not closely associated, but they tend to be nearby. So that might be the easy way in. People aren't afraid of geology. So like find a paleopodologist. And then once you get the paleopodologist, like, well, why don't we also look for leaves over here? Because yeah, that's how you really get the full picture. Because I find it fascinating that people can say that like, oh, there are primates here. Therefore, it was a closed environment. Like, whoa, 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 whoa. Yeah, you're making bold statements there. But yeah, when I was in Kenya, I was basically like, okay, so now all of you know a paleobotanist. So I expect to get a call because y'all need a paleobotanist on everything that you're doing. Because yeah, I think that basically the more people, the more eyes that you can get on something, the better. Because I'm learning as the paleontology collections manager in charge of animals that like I don't know what things look like because it's not what I study. And so it goes both ways. Like if you don't study plants and you're presented with one in the field, you might be like, that looks cool. I don't know what it is. Use the buddy system. The science needs to use the buddy system more.
Got to put that on a t-shirt. Here's a good question from Fossil Detective. Can you ever find fossil communities where plants look ill or sick that suggest that they were in stressed environment or a climate they didn't quite like?
Okay, so I don't know of any evidence for this. And part of the reason for that is probably because, so one of the things we talk about in paleobotany is that if a plant is not reproducing, it doesn't live there. So in general, like there are palm trees that live in Vancouver. They're probably not very happy trees. They probably wouldn't make it very well into the fossil record because if you think about it, like if you are nutrient stressed, you're probably not making do plants have bodies? I'm going to say they do. If you're a plant that's stressed, you're probably not making the best body that you can, and it might not preserve so well. But I don't know. That is not exactly something that I look at. I do know that I'm trying to think of like the closest I could find to a stressed plant. I don't know, ones that have been eaten. I betcha though that if you got brave and looked into like the isotopes, that might give you a better sense. Like, oh, this is a very sad plant. But yeah, I wonder. I wonder if sad plants make it into the fossil record, or like communities of sad plants make it into the fossil record. I have no idea. That's a good question.
So do you
Well,
think?
like,
So wait, does that mean that my one of my sad house plants won't make it into the fossil record?
Yeah, I doubt it. I'm sorry. Well,
Aw.
I don't know. Humans increase the preservation potential of a lot of things, so
well,
maybe.
talking about that, like on more of a conservation background, like the way we've really just taken plants and taken them in environments they shouldn't belong, and like the whole thing about invasive and
Mm-hmm.
planting native species, like how much do you think like we have messed up like the floral paleoecology of like
Yes.
things?
Oh my goodness. Okay, so one of the things that has been blowing my mind recently is okay, so I call myself an applied paleobotanist. Like I spend a lot of my time thinking about modern plants. And one of the things that has blown my mind is I am from Michigan. Like I am very familiar with the plants that are supposed to be in Michigan. And every so often I will go to Wikipedia or whatever and I will look at the actual distribution of these species, and it's nowhere near Michigan. So, like, for example, I always thought of Osage orange as being like, oh, that is a tree that you find in the eastern deciduous forest. Like, it's a thing. I found it wild in the forest in Michigan. No, it's native to like northern Texas and southern Oklahoma. That's it. It's like, are you serious? Like, this has happened to me multiple times. That just because a plant can live somewhere doesn't mean it's part of its range, and that is part of because of the ways that like plants um, disperse, right? Like they got lots of creative ways of doing it, but like you have to stay still and your offspring go out. So, it limits you in some ways. But I mean, humans are great at moving plants. We do it all the time. But that's also a thing that needs to be considered because there are other ways of doing like paleoclimate reconstructions where they will use the nearest living relative approach, and that can be super dangerous depending on one, how old the fossil is that you are like bringing forward to living relative, but also like where'd you get that data about what is the like climate tolerance of this plant? Because if you think about it, ginkgo, which is everywhere now, is super like its native range is like right here, this mountain. And there are lots of groups that like today there is one modern relative, but like is that in any way representative of what like all of these species were like? And yeah, especially considering that like we are very aware that ginkgos can live in many other places, but like are they happy? Are there these happy ginkgos? I don't know. It's dangerous. It's a dangerous game.
I remember reading a book, I can't remember the title, where it's like they were talking about how humans co-evolved with plants, and like at one point that it took like a weird turn where plants, this is describing it poorly, but plants are using humans as like a way to disperse themselves, and like we've become like the factor for dispersing plants and extending their range and stuff. So at what point do we like consider us taking plants and putting them in other places outside of like the natural quote order of things, right?
Right. Well, and like invasive is so loaded, right? It's this term that like in conservation means something very distinct. But like in terms of like the fossil record, that that's these are just range shifts a lot of the time. Humans just happen to be really good at ramping that up. And yeah, at what point is this like we're just part of the dispersal method? Like these invasives, no, 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 we're just really good dispersers. And I don't know where that line is because it's complicated. Plants make everything more complicated.
Yeah, they do,
Goodness,
and they give
that's
me headaches too. They're like, "Oh, I'm gonna water you with tap water today," and they're like, "Nope, I'm gonna die now."
Yeah. Well,
goodbye.
and it's so funny because like golly, plants are just the most like they're such weirdos, and it's one of those things that like we think of them. Oh, you don't move. You don't have to like catch your food. It's so simple. It's like no, plants can just be like I'm doubling my genome today, and like it's just a casual thing that they do.
Quick
Oh.
question before we go move on. as a collections person, right? So now you're trying to organize animals and plants, but like when, as you know, when we're organizing vertebrate collections, there's like ways you can do that based on like time, place, taxa, or like the type of animal it is.
Mhm.
With plants, because sometimes you just have like a leaf impression and a shape. How difficult is it to curate and catalog and organize a plant collection?
Yeah, it's a different beast entirely. So I ran into that. I ran into that during my dissertation because so the fieldwork that I did, all of the leaves that I collected are now in the National Museums of Kenya collection, and I personally doubled the size of the plant collection at the NMK because I filled a Land Rover with leaves. I'm very proud of that. But anyway, it was an issue just because like the collection staff had never had to like basically had never had to deal with it, especially not on the scale that I was doing it. Because you're right, I don't work with species. I work with shapes. And how do you organize that? And so by and large, in my experience at the NMK and here, mostly it's by locality. It's much more like stratigraphically oriented just because like leaves are the worst way to identify a plant. Like they are the worst because of all of things that make them great for reconstructing environment. It's terrible for identifying them. So if you have like fruits and flowers and seeds, oh yeah, you can use those. But like, yeah, basically leaves can be like, it came from this place, and that's really the most that I would try to do with it.
Oh man. All right. So I have another question, and
right.
it's I think it's like the hardest question we tend to give all of our Fossil Friday chat speakers. What's your favorite plant?
Okay. The funny thing is, people always ask me this, and I'm still never prepared. Okay, I guess.
So when you start forgetting like every plant you've ever
Right.
known,
And
like I know nothing.
I don't know a plant, but I need to be honest. Like I need to admit that I do know what my favorite plant is, and it's acacia.
Ooh.
I can't even like this is embarrassing. If I am watching like a TV show or a movie or whatever, and there are acacias, I have been known to just burst into tears because I think they're so beautiful. Like, I love them so much. They have these huge spines. Like, they are weapons. I love them. Like, they don't mess around. And they just have this, like, absolutely beautiful, like, distinct shape. And yeah, when I went to Kenya and actually saw acacia, I cried. I went on a bird tour to Nairobi National Park. So we were supposed to be looking at birds, but also like, rhinos and whatever. And our guide was a trooper because he'd like point out all the birds and I'd be like, what tree is that? What tree is that? And he knew, which I appreciated, but like, I was like, the birds are really cool. But also, what's the tree they're sitting in?
I would be the same if there were like ferns. I love ferns like crazy.
Yes. They're so pretty. I have the I still call it the lap fern, even though I took it with me when I graduated. This poor thing that like it's not happy, but it won't die, which is great. It got neglected when I went to Kenya for 2 months. Like nobody watered it, but it came back. Like this is a tough plant. Kansas nearly killed it because it gets really dry here in the winter, and I didn't think about that. It was just like help me, but it's looking much better now.
I love ferns, but I live in a desert, so it is difficult. So I have a particular affection for aroids. I think they're neat.
I support this. I support this.
In our chat, there are a couple folks who are like cacti versus euphorbia, so we'll see how that goes.
So one of the things that I recently learned, so there's basically it was always like, oh yeah, euphorbia is like Africa and you find cacti in like North America, but recently there is one single species of cactus found in Africa.
Ooh.
Really?
And so it just ruins that generalization, which I love. Like plants, they're so complicated. I love it. But yeah, no, I think because I work in Africa, like I have to be team euphorbia, but like I have been known to get way too excited about prickly pears and just like I have I was in Colorado and I met a cactus and I was like petting it and I kept getting stabbed by the spines and people are like, what is wrong with you? It's just such a pretty plant.
I was saying in the chat, like, in the end, plants are just the best in general. So
Yeah.
we'll go with that.
Yeah.
Well, I think that about does it, huh, Britt?
Yep.
All right. Well, thank you so much, Aly, for joining us on the show today. That was really, really great. And as you can tell, we're a bunch of plant people. So we were very, very happy to finally have plants on the show. One question that we kind of leave everybody with is, if you had advice for a future paleobotanist or someone, what would you say to them really quick?
Oh, that is a really hard question. Basically, I think the thing that has helped me the most is don't not ask a question because you think it's stupid. Like asking these very like things that are, oh, that's a really simple question. Maybe it's not. Like maybe it's not. So don't be afraid to just like start asking questions. But at the same time, honestly, the thing that I think helped me most as a scientist has been knowing when to say, I don't know the answer to that. I could tell you like, this is what I do know, but I don't know the answer to that. And I think that that is like, not being afraid to ask questions and being able to admit when you don't know something is a really good way to kind of grow and mature as a scientist.
Great advice. There's definitely many times where like, I have no clue. Sorry.
Yep. Same.
If anybody wants to like learn more about your research or the Sternberg Museum, where's the best place for them to go?
Alrighty, lots of places. Okay, so I am on Twitter and Instagram as Paleolorax. The Sternberg Museum is on Twitter and Instagram at Sternberg Museum, which is very nice. I also run the Fort Hays State University Paleo account. So that's on Instagram and Twitter at FHSU_paleo in both places. And then in addition, if you like seeing my face and listening to me talk about plants, the Sternberg Museum also has a YouTube channel, and I am on there periodically doing videos about why plants are really the thing that you should be excited about. So yeah, there are plenty of places on the internet where you can find me. I live on the internet.
Awesome. Well, I'll be sure we'll subscribe right away because more
Yay.
plants, yes. And we'll put all the links to those in the description below. That does it for today's episode. Thank you all so much for tuning in. Really quick announcement. Starting from here on out, we are very excited to say that Fossil Friday Chats is now partially funded thanks to a grant from the Paleontological Society. So thank you so much to everyone at the Education and Outreach Committee at the Paleontological Society to help us continue to tell these amazing stories from the world of paleontology. We'll be back next week with Ray Troll, who'll be talking to us about Cruising the Fossil Freeway, I think it was the name of his book. Be sure to like and subscribe, and we'll see you all next week. Bye, everybody.
Bye, everybody.
Bye.